Okay, so here we are to talk about genetics. So when we talk about genetics, we're going to start to figure out how, or try to figure out a little bit about how we inherit all of our traits. And there's all sorts of traits, and all of them are genetic. So we're talking like physical traits, like, our height, our shape of our face, all that kind of stuff. Biochemical traits, which might be our susceptibility to specific diseases, and our behavioral traits, like if we're extroverted or if we're shy. All of those sorts of things are heritable, right? We carry all that information in our genes, and of course we inherit that from our parents. So there's quite a bit of terminology that we have to sort of get straight before we can get too excited about genetics. So again, a gene was the thing I talked about at the end. A gene is essentially the basic unit of information that affects each and every genetic trait. If we were to look at this at the molecular level, a gene is basically a stretch of DNA found on a chromosome that is found in the nucleus of our cell, and that stretch of DNA codes for the production of a protein, a specific protein, and or a nucleic acid like RNA. So when we think about our traits, there's a couple terms that we need to know. The first is genotype. So our genotype is essentially our genetic makeup, right? It's the actual makeup of our alleles. So if every cell is diploid, then we get 2 alleles per gene, 1 from each of our parents. So we inherit these. This is not something you can see when you look at a person necessarily, right? This is something that we have to kind of, if we're trying to figure out the genotype, we'd have to sort of work with what we know, which is what we can see. So if we inherit 2 alleles from each of our parents, they can be the same, in which case we would say we are homozygous for that trait. Homo meaning same. So homozygous means that the alleles that you inherit are the same. They are either both dominant or both recessive. So a dominant and recessive genes are represented by capital letters and lowercase letters. So the dominant genes are represented by capital letters, and the recessive genes are represented by lowercase letters. So if you inherit 2 recessive genes, you are said to be homozygous recessive. If you inherit 2 dominant genes, you are said to be homozygous dominant. If, however, you inherit 1 dominant gene and 1 recessive gene, you are said to be heterozygous. And when you use the term heterozygous, you don't put recessive or dominant with it because by the nature of being heterozygous, that means you have 1 dominant and 1 recessive. So you want to be as specific as possible when you can. So you would never, for example, if you were given a test question and you were asked to give the genotype, you would never just write homozygous, because you need the either homozygous recessive or homozygous dominant. But if they are a heterozygous, meaning they have a dominant gene and a recessive gene, then you could just write heterozygous and that would be fine. So again, your genotype is your genetic makeup, meaning you have 2 dominant genes, 2 recessive genes, or 1 of each. Now that sometimes, if you know what trait you're looking for, and if you know what you're seeing is recessive and somebody shows it, then you can guess that they're probably homozygous recessive. But for the most part, you can't see the genotype. You can, however, see the outward expression of the genotype, or the physical expression of their genetic makeup, and that's called the phenotype. So for a particular trait, a genotype is the pair of alleles that codes for that phenotype. So in this example, we have the phenotype being tall or short, in which case if somebody is tall or if they're short, that's what you see. But you don't see their alleles for those traits. That's something you would have to know a little bit about, which is the dominant trait, which is the recessive trait, etc. So these are just terms we're going to come back to. So genotype is the genetic makeup, or what you get in terms of, that's how I remember it, what you get with a G, what you get in terms of alleles. Phenotype is what you outwardly express, or what you present to the outside world with a P, present for phenotype. That helps you to remember. So phenotype is what you see, and genotype is what you inherit. And sometimes you can know the genotype if based on what you see, but not always. All right, so that gets us to Mendel. So Mendel been sort of coined the father of modern genetics. And he did his work in like the middle 1800s. And he's very famous for breeding pea plants in his monastery garden. And through his work with pea plants, he discovered a lot about inheritance that scientists have continued to expand on, of course. So they're called Mendelian, it's called Mendelian genetic, or Mendel's laws of genetics. Is how we refer to this. There are some traits that are inherited via these Mendelian laws. Unfortunately, with humans and animals in general, it's not always that straightforward. But we start with the easy one, and Mendel's work is the easiest to understand. It's kind of the most straightforward. So just to get you up to speed on what he did. So you got to remember that this is in the mid 1800s, right? We've never seen a cell before. We don't really know much of anything about what's going on. Nothing about genes, nothing about DNA. None of that has been discovered. So this is pre all of this. And he was trying to figure out, curious about inheritance. And so like I said earlier, he was breeding these pea plants. So what he noticed is that each time he bred pea plants together, he was looking at the outcome. So basically, when he was breeding these two plants together, he was performing what we refer to as a genetic cross. A genetic cross is essentially a controlled mating activity that is done to examine how a specific trait might be inherited. So he basically did quite a few of these genetic crosses. And so if you look on the slide here, this is where we start. The P generation stands for parental. So he started with what we call true breeders. And a true breeder is a plant that when you breed it with another plant of the same flower color, the offspring always had the same color as the parents. So those are his true breeders. And so he had true breeders. And so he did this and looked at a couple different traits when he was doing this. The first thing that we're going to focus on are the seeds. So in his parent populations, he bred two types of plants that had yellow seeds and plants that had green seeds. And prior to that, when he bred yellow plants with yellow seeds, pea plants with yellow seeds with other pea plants with yellow seeds, he had yellow seeds. If he bred pea plants with green seeds with other plants with green seeds, he had green seeds. So then what he did is he took the yellow parent, one of the parent plants had yellow seeds and the other one had green seeds. And so he wanted to cross them to see what happened. And if you just look to the bottom, you notice in his first generation, the offspring all had yellow seeds. So that got him started, right? That there's something going on here in terms of one, you start with yellow and you end with green and the offspring have yellow seeds, then that tells us that yellow dominates over green, because the yellow is what we're seeing in the generation, in the offspring. So that's called a genetic cross. He did this also with flowers, and we'll look at flowers in a minute. We'll have another one with flowers. He did purple flowers and white flowers, and I'll show you that. So he was looking at flowers, he was looking at seeds. So you kind of focus on your trait of interest. And in this particular slide, the trait of interest is the seeds. Okay, so the first generation is the F1 generation. The F stands for filial, which is a word that refers to son or daughter. So then what he started doing, then he started crossing the individuals of the F1 generation to see what happened. And the offspring were then the F2 generation, and so on and so forth. That's sort of what he did. So in order to predict the results of these test crosses or these experimental crosses, we can use a grid-like diagram called a Punnett square, which you can see there. And you probably maybe did this in biology. So a Punnett square shows us all the possible ways that two alleles can be brought together when we have fertilization. So to create a Punnett square showing how a trait is inherited, what you have to do first is you have to list the alleles on the outside of the grids, if you will. So basically, we have for our F1 generation, he mixed the sperm and the eggs, and in this case, what we see is we have heterozygotes. We have a dominant gene and a recessive gene in one of our parents, and we have a dominant gene and a recessive gene, in this case for seed color, in the other parent. So this is the F1 generation. This is the first generation from our true breeders. So, and we're looking at only one trait. So it's called a monohybrid trait because we're only tracking one thing, and that's seed color. So when we did this, we have the eggs. So this is the mom, this is the eggs. So we split the alleles for this one trait, right? We have two alleles for each trait. So you split the alleles, you put a dominant here, a recessive here. You put a dominant here for the sperm and a recessive here. If there were two dominants, you would put a dominant and a dominant. If there were two recessives, you would put a recessive and a recessive. So when Mendel did this, he saw that with the monohybrid cross of the heterozygotes of the F1 generation, he ended up with, so how you do the sort of filling in the Punnett square is you just fill in each box. So you take the Y here and you drop this Y here. So we have two, this is a homozygous dominant, yellow-seeded offspring potential. Then we take the big Y here and the little y down here, which also turned out to be yellow seeds, but this is a heterozygote. Then we take the big Y, you always put the capital letter first. Doesn't matter. We always list the capital letter first. So we bring the capital Y here and the lowercase y here for another heterozygote. And then finally we bring the two recessive, little y, and that one turned out to have green seeds. So the recessive gene is the little y. The dominant gene is the big Y. Any offspring that inherits one, all you have to have is one dominant gene in this type of inheritance, and you'll show the dominant trait. So 3 of the offspring showed the dominant trait, which is yellow seeds. 1 of the offspring shows the green seeds, and that one's the homozygous recessive. So our ratio is, in terms of what we can see, our phenotypic ratio would be 3 to 1, 3 yellows to 1 green. If we wanted to do a genotypic ratio, it would be 1 to 2 to 1, 1 homozygous dominant, 2 heterozygote, and 1 homozygous recessive. So that's what happens when you, and that's predictable in this kind of inheritance, in what we refer to as Mendelian inheritance, when you breed heterozygotes, for a specific trait, you're going to always have that characteristic ratio, phenotypically 3 to 1. Hopefully that makes sense to you. So let's look at this in some words. So based on this experiment right here that we were just talking about, what he kind of observed and came to the conclusion is there wasn't any blending of the trait, right? It was yellow or green. It wasn't like yellowish green. So that means that if your options are either yellow or green, that means there's a dominant trait and a recessive trait. In order to show the dominant trait outwardly, phenotypically, all you need is one dominant gene in this particular type of inheritance. To show the recessive trait outwardly, phenotypically, you need to have two recessive genes. You must be homozygous recessive. So he didn't call them genes, he called them elements because he didn't know anything about DNA at that time. But he knew enough after his series, and he did this a lot. I don't remember off the top of my head how much, but it was like a lot. You have to do this, I think he did it like almost like 1,000 times roughly. And, he had a lot of patients, and he kept getting the same kinds of results. So, based on that, he learned that these genes come in pairs, or these elements come in pairs, and that only one allele is in each gamete. So you don't get two alleles; you get only one allele in each gamete, and he called that the law of segregation. So, his results basically led him to this, which he called the law of segregation. So, in modern terms, the law of segregation would state that two alleles of a gene are separated during meiosis, which we know. We just talked about that. So two alleles of each gene are separated during meiosis, and each of them get into the gametes, right? And so this particular law of segregation, this understanding this understanding that the alleles are separated, the alleles for each gene. Now think back to what we were just looking at. Remember how we saw those paired homologs? How when you have them joined together, look back at that picture where you see the blue and the pink ones, where you see A, B, C, D, E on both arms of those sisters. Those get separated. That's what we're talking about. The two arms of those chromosomes that are joined together, each of those sisters each had A, B, C, D, E, and then they get separated. So that's what he's talking about here. So you inherit just one of those from each of you inherit one allele from each gene for each parent, because the product of fertilization is the pooling of the genetic information. So the A and the A come together when fertilization occurs. All right, so this law of segregation can basically be used to predict sort of this genetic outcomes in this particular type of cell division. So this is what we were talking about, right? So the law of segregation, if this for a particular gene, if this was in the parent, ultimately these alleles would separate. By the end of meiosis 2, these alleles are separated in the gametes. So that's the law of segregation. So there it is in words. When the diploid cell undergoes meiosis, homologs separate, we know that, and the two alleles of each gene are packaged into separate gametes. During fertilization, the gametes come together, and they carry one member of each homologous pair. Okay, so let's pretend to be Mendel and not have any of this modern tool that we have, these tools now. We have now these DNA sequencers that you just run it through a machine and it spits out everything. That he didn't have the luxury of that, and he didn't know anything about DNA at all at this point, which is kind of amazing that he got as far as he did. So how could we tell if an organism that shows a dominant trait is homozygous dominant or heterozygous? So think about that for a minute. How would you go about trying to decide that? If we have an organism that's showing us a dominant trait, we can't tell by looking at them whether they're homozygous dominant or they're heterozygous, because you only need to have one of those big T's to show the trait in this particular type of inheritance. So if you're thinking about it, you might say, well, if I could cross it with something I know for sure. So that's what you do. You form a test cross. So if you're trying to figure out if in this case, the gene that we're looking at is for being tall as a plant. Being tall for a plant is a dominant gene. So the plant will be tall if it is homozygous dominant, like you see right here, big T, or if it's big T, little t. Both of these plants will be tall. So you can't tell by looking at a tall plant if it's this or this. So if you wanted to figure out, is it this or this, how do you go about doing it? Well, you cross it with something that you know, and that would be the homozygous recessive. If you have a short plant, it has to be little t. So you take a male plant that's little t, and you cross it with your female. which would either be big T or big T little t. We don't know. So basically, you would draw your Punnett square and you'd figure it out. So if your female plant was big T, and your male plant, the known is little t, all the offspring, so you'd fill it in, little t, big T, and it doesn't matter if you put the female up here or the female down here, just if you just do it the same way every time, it doesn't matter. So you notice that all of our offspring in this case would be tall. They'd all be heterozygous, but they'd all be tall. Versus if the female was big T, little t, half of the offspring would be tall and half of them would be short. So that's how you would have to go about doing this. If you were Mendel and you didn't have any other way to do it, you'd have to do a lot of experimentation. So you'd mix your plant over and over and over again with a known and see the ratios, right? So and again, if you just did it once, you still don't know, right? You have to do it a lot of times. This is just probability, right? It doesn't say for sure that here, for sure, all the offspring are going to be tall. You could do this over and over and over again with these two parents and they're all going to be tall. But if you only did it one time and you got a short one, you got to do it a bunch of times to really get the ratio, to really get the odds. It's just an odds ratio. It's not a for sure, because it's a flip of the coin every time what you're going to get. But that gives you a probability, basically, is what we're doing. We're giving we're just predicting the ratio and giving a simple probability that those particular offspring will have a certain phenotypic or genotypic expression. So it's odds. Okay. So then we have if we make it a little bit more complicated, if we're looking at two traits, we would call that a dihybrid cross. So a dihybrid cross is studying two traits. In this case, we would be looking at the color and the texture of the seed pod. So the color of the seed pod, the recessive color is green, the dominant color is yellow, the recessive texture is kind of wrinkly, and the dominant is smooth. So this makes it a whole lot more complicated in terms of the Punnett square. But what we're trying to figure out here with the dihybrid cross is if the inheritance of one trait influences the inheritance of a second. And in humans, this is very often the case. So what he wanted to figure out is if that was the case in the peas. And so he had, this is what he started with. And he started with a homozygous recessive for both traits. So we have yellow and smooth with a yellow and smooth, and then they just predict the offspring. So this is kind of how this shook out. So this is called a dihybrid cross, right? And it's bigger. This Punnett square is a lot bigger because you have to give all the possible combinations for this one. So your possible combination would be little r, little y. So there's one, big R, big Y, little r, little y. Big R, big Y for here, little r, big Y for this one, and big R, little y, right? So every possible configuration for this particular parent, and then the same for the other parent, and then you do your square. So when you look at this, we have a situation where we have our offspring are going to be either green and smooth, yellow and smooth, wrinkly, or I should say green, yellow, wrinkled, and smooth. And so when you look at our offspring, we have, in this case, only one full homozygous, heterozygous dominant, or heterozygous recessive for all of the traits. So we've got a green wrinkly. We've got a bunch of yellow, smooth, 1, 2, 3, 4, 5, 6, 7, 8, 9. We've got 3 yellow and wrinkly, and we've got 3 green and smooth, right? So it's and there's 16 total. So essentially 9 out of the 16 are round and yellow. 3 out of the 16 are round and green. 3 out of the 16 are wrinkled and yellow. I'm using round and smooth for the same. And 1 of the 16th is wrinkled and green. So basically that's a 9 to 3 to 3 to 1 ratio. Don't need to remember that. But it's telling us that what the point of this part of me was to test the hypothesis that the alleles of 2 different genes are inherited independently from one another. And they have to be independently from one another because they are spread out all over the place, right? There's such a mix, I guess we could say. So that gives us an idea that traits controlled by a single gene and unaffected by environmental conditions, we call those Mendelian traits. So again, you kind of have to think like he didn't know anything about anything. So in terms of DNA, right? This was all sort of observation. All right, so this law was called independent. So the first law was segregation. This law is called Mendel's law of independent assortment. And it states essentially that when gametes form, the 2 copies of the alleles of any given gene segregate during meiosis independently of any 2 alleles for other genes. So in our example, pea seeds could be round or wrinkled, and they could be yellow or green because those genes are basically controlled by different traits, right? So the smooth or the round versus the wrinkled, R gene controls the seed shape, and the Y gene controls the color of the seed, and neither one affects the inheritance of the other. That's essentially the big difference. If the genes were on the same chromosome, then they would sort together, right? If the genes were right next to one another on the same chromosome, that one chromosome is going to separate, but the genes that are housed on that chromosome are going to be inherited together. So he just happened to get lucky in that he chose genes that weren't inherited together. It would have been a lot more confusing if he would have chosen genes that were right next to one another on the same chromosome. But luckily, that wasn't what he did. All right, so there was a lot of luck involved in this for this first time. All right, so it's not always dominant versus recessive. There are some other things that can confuse this a little bit. And where he saw this was when he started looking at flower color. And flowers, it turns out, show what we call incomplete dominance. So you notice that we have red flowers being big R, white flowers being little r, but when he bred them, some of the offspring were pink. So the pink is the heterozygous. So basically the red doesn't necessarily dominate it's not complete dominance over the white. It's sort of like a blend, right? It's sort of shared. So if the heterozygote is pink, that means that this is a blending of these genes, right? So they're both expressing to some degree. And so you can see that when you do their test cross, what we crossed here was 2 heterozygotes, which would be two pink flowers. If you cross 2 pink flowers, 1 of the 4 potentially would be red, 2 would be pink, and 1 would be white. So it's still our 1 to 2 to 1 genotypic ratio, but in this case, phenotype, we've got the phenotypic ratio would be 1 to 2 to 1 as well. 1 red, 2 pink, 1 white. So that if you see that, this sort of blending, this third choice, you would think about that being called incomplete dominance. All right, let's look at another example. So this is called codominance. Now with codominance, the heterozygote is going to fully express 2 different alleles at the same time. And a good example of this is blood typing in humans. And we'll do a lab on this a week. So with blood typing in humans, our human blood types type choices are type A. You can look at them down here. Type A, type B, type AB, this is the codominant one, and type O. How this is determined is essentially by what kind of proteins are on the surface of our red blood cells. So basically, the gene for blood type has 3 alleles. It can be there's 3 alleles. There's A, there's B, and there's O. But A and B can be expressed at the same time. That's the codominant part of this. Right, so if you look over here, so this is a person with type A blood has these A antigens on the surface of their blood, these A proteins on the surface of their blood. If you're type B blood, you have B proteins on the surface of your blood. If you're type AB, you have both A and B proteins on the surface of your blood. This is the codominant one. They're both being expressed in the offspring. And type O has no proteins on the surface of their blood. So how we write this is blood typing. We always write a big I and then a superscript letter if it's A. So if you have type A blood, your choices are to have inherited 2 of these genes from each of your parents. So you would be big I, big A, big I, big A. Your other option would be to inherit 1 A from one parent, so big I, big A, and then if your other parent was an O, you would inherit the little I, the recessive. So you would be big A, big I, big A, little I. Either one of these would give you type A blood. If you're B, your choices are big I, big B for being homozygous dominant, or big I, big B, little I, heterozygous. Still type B blood. And it's because the I has no surface antigens. They don't have anything to add to that. However, if you're AB, you have both A and B. You inherited from one parent a gene for type A blood, you inherited a gene for type B blood from the other parent, and you express them both. That's codominance. And then if you're O, you're recessive. You have no surface antigens, and you would have inherited a gene from 2 recessive genes from each of your parents. So we have the codominant and the recessive. And this is called codominant because in the AB offspring, person that has an AB phenotype, they have both the big I, big A allele and the big I, big B allele both express. That's called codominance. Versus what we just saw with incomplete dominance, they're not both expressing, right? It's a blending. It's an intermediate between the two, whereas this is full expression with type AB blood. All right, to get it even a little bit more confusing, there's something called pleiotropy. So now we're moving into more complications, right? A more complicated situation. So what we know is that lots of traits tend to be complex in that there's more than one gene at play. So a complex trait is one where we cannot really predict the pattern of inheritance by the laws that Mendel laid out for us. They don't fit that straightforward kind of single gene, single phenotype pattern that we've been talking about. In lots of cases, a single gene can influence a number of different traits, and that's what we refer to as pleo, because pleo means many and tropy means change. So pleiotropy means there's many things at play here. So this is an example. This is an example of a condition called Anderson syndrome, which there's one protein that plays a role in more than one biochemical pathway and therefore affects more than one body part if that particular protein is represented. And so this thing that you see here is something called a pedigree, which I'm going to talk more in a little bit. Pedigrees they're like genetic family trees, and the circles represent females, the squares represent males. When you fill it in, that represents a person being affected. And then you can see the sort of key. So they mean different things. So if it's filled in like half like this, that means just the muscle is affected. This would be structural. If it looks like this piece of the pie, this piece of the pie would be the heart is affected, and then a blank nothing would be nothing is affected. So when you see this kind of thing, that's obviously not straightforward, right? So that gives us an idea that this is pleiotropy, that there's more than one gene, pardon me, is influencing a number of different traits. And you can see this in all sorts of different organisms. I was reading in one of my biology books about they were doing a long-term breeding experiment in Russia to make their silver foxes tame. And they noticed that again, you have to do this repetitively. And as they started breeding these foxes that were tame together, the subsequent offsprings became more and more tame, but then they also had some other unique things, like their ears started getting floppy instead of straight, and their legs got shorter, and their tails got more kind of curly, right? And so that tells you that there's more than one thing at play, right? That's called a pleiotropy. Another thing that we see, that is even kind of more complicated, is when we have the more than most, and what we know about humans is most traits are probably controlled by the action of more than one gene, and these are called polygenic traits. That's coming next. Sorry. This one is sort of interesting. This is back with pleiotropy, but it's showing us some kind of a unique thing. And this is when environmental conditions can influence gene expression. And this is something we're learning a lot about, that the environment definitely plays a role on how our with respect to how our genes are expressing. And we're seeing this more and more and more the more we learn about genetics. But in the case of this little kitty cat here, this little Siamese cat, we know that temperature in these animals, and you see this in some rabbits too, temperature affects the pigment molecules, the quality of the pigment molecules in the fur of some animals. So where the animal is cooler, where it doesn't get a lot of circulation, like its little paws, its tail, the tips of its nose, its ears, their fur is darker, where their body is cooler. So the temperature affects, in this case, the pigment molecules. So that's telling us, that's complex, right? But it's showing us that environmental conditions can also play a role. And then this is what I was just about to talk about, was with the polygenic traits. So polygenic traits in humans, polygenic traits include all sorts of traits, like our eye color, our skin color, how fast we run, how big we are. Of the thousands of human genetic traits, probably 25,000 of them are governed by this type of polygenic inheritance. A much smaller percentage is inherited by Mendelian inheritance patterns, straight dominant recessive. The rest are probably polygenic. So it gets harder to talk about this in humans. And when you have a polygenic trait, oftentimes what you'll see, if you have a big enough pool, you'll kind of see this bell-shaped curve. And then I told you that environmental influences very definitely play a role. So here's sort of just a graphic of this polygenic kind of inheritance. And these are just looking at people, obviously, and just kind of the bell-shaped curve. So it's hard to see who's the men and who are the women in this pool here. But you have your small people right over here. This is about, I think this is the 5-foot mark, and over here there's the 6'2" mark, right? So you got, a couple, like one short person, about 5 foot, 5'1", and then, the bulk of them, they all lined up. It's sort of cool how they did this. They all lined up. This is the 5'5", 5'6", 5'7", 5'8", 5'9", and then we start coming down the other side. Sort of the similar presentation here. This is the 5-foot mark. The people in white are females. The people in blue are males. So we have, again, our sort of standard, like the most of them fall right in here, right in the middle, 5'5", 5'6", 5'7", 5'8", 5'9", and then we start going back down the other side. And it's the same kind of a thing for, in this case, they're looking at unit of pigment in skin and in hair and in eyes. All of those are all of those influence. All right, so that's those, and again, that is the most common type of inheritance in humans, is this polygenic, these polygenic traits. That's how most of our traits are inherited. They're influenced, the action is influenced by more than one gene. Okay, so that I'm going to stop here, and then the next video I'm going to talk a little bit more about human genetics. And again, in class, we're going to do a couple activities in class on Friday. We're going to do a mitosis meiosis, and I'm going to give you some a genetic worksheet to do.